going to take a couple minutes, and I don't want to eat up into too much of Minya's presentation time. So thank you all for coming. We are thrilled to have you all here on this very special occasion. It's always so much fun to see a student's defense after all that hard work. So I am just going to share my screen. Bear with me. Okay. So today we are here to celebrate Minya. So I'm going to tell you a little bit about her, sort of how we got to know each other. And the first thing I want to do though is tell you that contrary to what the impression you may get over the next hour, Minya does have a weakness. Okay, so she does not like insects, bugs, invertebrates. Like if you want to see her run away screaming, you know, show her a little inchworm or something like that. So I'm just giving you something to hold on to over the next hour as you are just completely blown away by her accomplishments and how amazing she is. Don't feel like she's completely, you know, untouchable. She does have a weakness. Okay, so I'm just going to give you that as a little bit of comfort. But now we're going to have a very short musical interlude. I'm going to see, and I apologize for the sound quality because I was having problems figuring out how to get this to work. So a couple of years ago, I was at one of Cassandra's students' defenses, and she always plays a song for her student that she specifically chooses for that student. And I immediately, as she was making this introduction, I immediately knew what song I would play for Minya. And actually, I already also knew what song I played for Molly, but we're going to wait a little bit longer for that. So I have her song queued up. This is a song by, oops, starting over again. This is a song by a band called OK Go. And the song is called Invincible. So I'm not going to play the whole thing, but you'll have to go listen to it on Spotify or something, because certainly the beginning of the song is how I think of Minya. I'm just going to fast forward a little bit. So it's about an alien invasion and the fact that this woman is the thing that they have to get through, and she is going to destroy them because she is invincible. So you'll have to go listen to that song, and then you'll understand what Minya has been like the last 6 or 7 years, because I've gotten to the conclusion that she can basically do anything. All right, so this is how it started. This is the first email that Minya sent me in 2013 expressing interest in doing a master's rotation in my lab. She was part of the MEME master's program in Europe, and I actually see some of her advisors and collaborators from that work in the audience, so it's wonderful to have you here. And I have to admit that I did not know what I was getting into. I thought, oh, she's obviously very smart and enthusiastic. I remember we had a Skype chat, and I thought, oh, it's 3 months, you know, sure, she can come. It's no problem. But I quickly discovered that Minya was really a very special student. As part of her rotation, she did the work that led to this first author publication. She also, I remember very well when she was here as a MEME rotation student, I said, well, you know, you could give an herbarium seminar, and yes, here's her irises. So you can give an herbarium seminar. And she said, oh, sure, I'll give an herbarium seminar. I said, you know, it doesn't have to be an hour. You can just do 25 minutes. And she said, no, I'll do an hour. And I thought, okay, all right. And I remember when I sat down with her to go through the talk beforehand, it was amazing. She gave this incredible talk about pollinator biology and the way pollinators see color and about irises. And I remember the day of the seminar, I walked in the seminar room, and John Dowling, who's a very famous vision biologist, was sitting there. And I said, John, what are you doing here? He's like, oh, I saw this, what looked like a great talk. And I came totally unperturbed. She gave her talk. It was fantastic. So that was my first sort of combination of the fact that, like, okay, she's got the whole package. She's great at research. She also is a great communicator. When she actually started a little bit early in the lab, and because we just gotten this grant, this was another first author paper she got. We hadn't even gotten to her PhD project yet. This second author is an undergraduate who she mentored for their senior thesis. This is the other senior thesis student that she mentored this particular year. So she's also a fantastic mentor in the lab. And what she's going to tell you about today, which I'm not going to go into any further, is what she actually did her PhD on, which is on floral meristem termination. So you'll hear a lot more about that. And she's also going to give you this kind of like trajectory. I'm not going to give you tons of her background because she's going to walk you through it. How did she come to work on this project? But I'm going to tell you a little bit more about some stuff that her colleagues have shared with me. So Minya is part of this group that I think of as the Snowpocalypse cohort. It was the largest group of students we've had in our graduate program. They're incredibly tight-knit, and so you're going to see a lot of them in these photos. In addition to being a wonderful scientist, Minya is also a very talented artist in drawing and photography. And here's, she made this cover for our G4 symposium the year that her class was presenting. And so all of these different images represent the talks that were being given by her colleagues, which was super fun. So she loves doing art and sharing her art with others. As I mentioned, she's a great speaker. She's a fantastic teacher. She's won multiple Derek Box certificates for teaching. She won award from the Botanical Society of America for the best student presentation in the development structural section. Here she is teaching some of her cohort Chinese and sort of like where the Chinese characters come from. Of course, she's fluent in multiple languages, including Japanese. She speaks some French and a little German. So yeah, again, like I said, I had to warn you, she's extraordinarily accomplished. She loves plants and she loves sharing her love of plants. So she's been very active with the Botanical Society of America and has won, again, multiple awards related to her work as an ambassador for plant biology. So she's always, you can always find her out and around New England and the world botanizing. Get past this. She loves to eat. One other weakness, so the insect weakness is something she's actually afraid of, but she also loves pistachio ice cream. She's not a huge sweet person, but pistachio ice cream is the one thing you can get her for, but she's a fantastic cook and we've all been able to benefit from that. This is a minor thing that came out to me when all of her friends sent me these photos. She seems to be very proud of the fact that she can roll her tongue. I had multiple photographs to choose from where she was rolling her tongue in the photo. So this is an unknown talent that I honestly hadn't been aware of before this. She's also extremely resourceful. This is a picture of her and Bengali Scott when she had rescued his phone from certain death in a storm drain over on the Science Center Plaza. So just in general, Minya has been a fantastic person to have in our community, in our lab. We're really going to miss her as she goes off to better things at the University of Connecticut at the postdoc. And yeah, so I think you're all just going to be thrilled to hear about her work today. And so I'm going to hand it over to Minya now.
Thank you, Elena. Mwah, mwah. Okay, everyone. Thanks for coming. Really happy to see so many familiar names and faces, and I actually don't mind defending on Zoom at all because at least half of you wouldn't be able to make it if it's held in person. So many of you here today have heard of me giving a talk or two before, and you know that I like dramatic openings. But today I will do something different. It will be a sincere talk instead of a theatric one, and I will start by talking about myself. I have been a student for way too many years, and during these years, I made some important discoveries about myself, and I want to tell you about these discoveries because they are the reasons that I am defending my PhD here today. So I started undergrad not as a biology student. I majored in material sciences, and in my freshman year, for example, I was learning all the things about what are the different properties of different metals and how to do technical drawings with a drafting machine. Later, of course, I managed to transfer my major to biology. However, this transfer of major was due to something extremely silly and personal that I'm happy to share over a beer. But the point I want to make here is that this is not because I disliked material sciences or I liked biology. I had no idea why I meant to major in biology, whether I would like it, what I would learn. I was completely ignorant. But things turned out to be all right after I started to take all the courses in biology. In fact, more than all right. After I learned things in molecular and cell biology, I honestly thought and still think that they are the most fascinating things in the world. Organelles, chromosomes, genes, RNA, transcription, translation, protein folding. I was obsessed with every little thing that I learned, and I felt I know the secret of the world. So after my undergrad, I decided to do a master's degree because I wasn't sure what I want to do as a job or a career yet. So one afternoon, I sat down and Googled master's degree, biology, scholarship, and Europe, because I wanted to go to Europe. Then I found this program that appears to be too good to be real, but still I applied, got accepted, and went to Europe. To date, this is still one of the best possible decisions that I can make in my life. But the plot twist for me is that that program focuses on evolutionary biology. While I have never taken any evolutionary bio courses in my undergrad, I thought it's biology after all, how different can it be? So with that very naive mindset entering that program, of course, I have experienced some huge culture shock. Everything was so new and eye-opening. Never ever have I thought that the genes that I studied during my undergrad would have sequence differences when they're in a moss cell versus in a sunflower cell. And the sequence differences can tell us a lot. It took me probably a solid year to come to understand and to appreciate what does it mean that nothing in biology makes sense except in the light of evolution. I find the process of adaptation and speciation very fascinating. Genetic drift, convergent evolution blew my mind, and I started to appreciate, to recognize the biodiversity that we have at our current timeline. Meanwhile, I made another discovery about myself, which is that I am a very visual and very organismal person. I always get very attached to the organisms that I study, and I pay attention to their structures and morphology. I enjoy all sorts of microscope work. That's how I decided to study plants, because I loved plants since I was little, but only by then I knew that all the attentions I paid to plants can have scientific values. What a win-win situation, right? Years later, I read a sentence from a book that goes like, we all must be morphologists before we can be biologists of any other sort. And that spoke to my soul. So until my master's, I had these three major discoveries about myself. But at that time, in my head, they are occupying very different spaces with very little overlap. One is looking at things at the smallest scale, the genes and the molecules and the precise mechanisms of their interaction. One is looking at things through deep time, reconstructing events that happened before at very large scale. And one is paying attention to just the individual or just part of the individual, and then appreciate their structure and morphology. So I thought I had to make a decision between them, and I was very indecisive. However, things took a drastic turn after I came to Elena's lab, which is also an opportunity that's provided by my master's program. The research going on in the Creamer lab shocked me because little did I know that there is already a field in biology that just perfectly combined everything that I like together. And that field is called EvoDevo, evolutionary developmental biology. What do I mean everything is perfectly combined? So the field of EvoDevo aims to understand what genes and genetic networks give rise to certain morphologies, how changes in these genes and networks give rise to morphological diversity, and what are the evolutionary consequences of such morphological diversities. You see, from genes to morphology to individual to evolution to diversity. What's more, the aspect of morphological diversity that the Creamer lab studies is extremely attractive, which is about flowers. So under this overarching theme that we want to understand the EvoDevo basis of flora morphological diversity, we can ask more specific questions. For instance, which genes control organ development in all flowers and which genes give rise to lineage-specific characters? What are the genetic mechanisms by which new flora forms arise? How do the key genes and networks in flora development evolve? And what is the genetic basis underlying the different flora morphs that led to or as a result of speciation? So after my master's, it was a very simple decision regarding whether I should join Elena's lab for a PhD. I will be gladly thinking about the evolutivo basis of flora form for the rest of my life. Since the flora forms that we see in nature is literally infinite, we always start our work from one system, Aquilegia, and then we expand our knowledge from there. As you can see here, Aquilegia has very charismatic flowers, and it's a very important model system as well. For the past 30 years, we have gained a lot of knowledge in Flora evolutivo, but the majority of this knowledge was obtained from studies from 3 major groups of plants. This information is incredibly valuable, but it's not comprehensive. Because our ultimate goal is not to understand how 3 groups of plants make their flowers. We want to understand what's going on in all flowers and what made them different. To this end, obviously adding more model systems at different phylogenetic positions will help, and our Aquilegia occupied a position there. So in my first 2 years of grad school, I got my feet wet, I got my hand wet, I got everything wet. I dived into lab and started with some candidate gene project. Candidate gene approach is a way to study the functions of genes in our system, Aquilegia, and then we compare what we know in Aquilegia to what we knew about these genes in other systems. Normally, it involves a few steps. So firstly, of course, I will have my genes of interest. And then I will need to build a gene phylogeny to confirm the homologies of these genes. Then I will need to analyze the expression level, expression pattern of these genes in different flora organs over as many developmental stages as possible. I will also make some RNA interference construct to silence these genes in our flower, aka make some mutants. Once these mutants bloom, I will then need to make some very careful characterizations regarding whether or not and how do those mutants look different from the normal flowers. What have changed and what caused these changes structurally? For instance, if there are some organs missing in the mutant, is it because those organs are very underdeveloped so that I wouldn't see them with my naked eye, or because they just didn't initiate at all? If some of the organs are smaller in the mutants, is it because it has fewer cells or all the cells are smaller? So all these characterizations involves all sorts of microscope work, and they are crucial for interpreting the functions of the genes. Based on our interpretation of the functions of the genes, we will then design the next step to push the investigation further. For instance, we can analyze the expression levels of other related genes in the mutant to see how they change. I thoroughly enjoyed all the candidate gene approach project, and I produced some results that I'm quite proud of. But more importantly, these projects really helped me to understand and to think about how to examine gene functions in the context of development and evolution. Meanwhile, I started to think about what kind of developmental processes that I can dive in for the rest of my PhD. You see, candidate gene approach is important. It's necessary. It provides us with direct evidence of what's the functions of the genes, but it's not comprehensive. It's a targeted and comparative approach, meaning that we'll have to get the genes from somewhere. If we imagine the developmental process that we want to explore is an entirely dark space and we only have one flashlight, candidate gene approach is when we have already gathered enough information so that at least we know which direction to point our flashlight to, and then we collect information along the way in that direction. So it's a relatively downstream process. What I wanted to do is to set up my own exploratory dark space, really explore it, and come up with some directions on my own. While going through the current literatures of Flora evolutivo, I became increasingly obsessed with the earliest phases in building a flower. When I say earliest phases, one would normally think about when the flower buds are very, very small. But no, I'm thinking about things that happened way before that. All flowers started from just a dome of cells, the flora meristans. So this dome of cells looks like nothing special, but they have a group of stem cells in the center. These stem cells, they would divide to maintain their own undifferentiated population, but they will also divide to give rise to cells that will differentiate and build into organ primordium. Domes of cells like this, the meristems, are in fact the foundations to how plants grow. For instance, the vegetative meristems produce leaves, and they can theoretically produce leaves continuously throughout the lifespan of a plant. But the story doesn't end there. So for each leaf that's produced by this meristem, they each subtend a meristem as well. When the plant feels like it, when the condition is right, any of these meristems can just start the process again, produce more leaves, and any of those leaves will subtend more meristems, and the cycle continues. This is why stem cell research in plant sciences is a fascinating field, but it's not nearly as vast as stem cell research in animals, because we don't have to spend a lot of effort looking for them or trying to activate them. When we look at a tree, we will know that stem cells are everywhere all over their tree, and they are constantly active. This is the foundation to how plants grow. This is also why if we break an arm, it doesn't grow back. But if you break a branch of a plant, a new branch will grow out and everything will be just fine. I personally strongly discourage anyone to just break a branch of a plant, leave the plants alone, thanks. But I hope you are impressed by this very different, very novel, very amazing way that plants have invented to grow and to reproduce. Speaking of reproduction, let's come back to the flora meristems. So instead of producing leaves, the flora meristems produce floral organs that will compose into flowers, which are the reproductive structure of most plants. Now, think about a flower. Probably you're thinking about something like this. So a typical flower has 4 types of floral organs, and they are arranged in specific positions. From the outermost position to the innermost, we have sepals, petals, stamens, which are the organs that will produce pollens and make us sneeze, and carpels, which are the organs that have ovules that will make seeds. So from a dome of cells like this to a flower like that, the very first steps are to produce all the flora organ primordia by the flora meristem. Flora organ primordia are produced in spirals or whorls from the outermost whorl to the innermost whorl. Once all the organs are produced, they undergo organ elaboration, changing their size, structure, and color, and grow into the flower that we see. However, here's something really important I haven't told you about yet. We all know that every flower only has a finite number of flora organs. No matter it's 5 or 5,000, it's a finite, it's a defined number. And that means the flora meristem cannot just keep producing organs forever. So at some point, the stem cell activity in the flora meristem will be turned off, and all the cells in the center will differentiate. This process is called flora meristem termination. I hope that by looking at this diagram, you can recognize that flora meristem termination is a universal process that happens in all flowers. It is in fact one of the defining features of a flora meristem, because unlike the vegetative meristem, a flora meristem will always be terminated. It's an important but also fine-tuned process because it has to occur at a specific developmental stage to ensure the correct number of healthy primordia to be produced in the flower. Moreover, this is also one of the most fundamental, really important sources for generating flora morphological diversity. If a flora meristem is terminated early, fewer whorls of organs in one flower, but if the flora meristem stayed active longer, more whorls of organs in that flower. For each flora form that's produced here by the variation in the termination process, they each can have their subsequent modifications in their flora organ structure, organ position, or the number of organs per whorl. And the interaction between all these processes together give rise to infinity. Surprisingly, though, we currently know very little about what's the molecular basis underlying flora meristem termination. Our only and very limited information come from studies in Arabidopsis thaliana, which is the E. coli, the yeast, the fruit fly, the mice, the human in plant sciences. So it has a very small flower with only 4 whorls of organs. Each organ type occupied 1 whorl. This super basic structure is good because it provided us with a good starting point of looking into this question. However, 4 whorls of organs in a flower by no means can represent the diversity that we see. Currently, we have discovered a lot of mutants in Arabidopsis that either have their stem cells proliferate in an uncontrolled fashion or have their stem cell activity completely eliminated. But we don't know how is the termination process fine-tuned to generate flowers of different whorls, but still stay as perfect flowers. For instance, what's the difference between a flower with only 4 whorls of organs versus those that have 10? We have no idea. Our lack of understanding in this very important process is due to the fact that Arabidopsis and other model systems all belong to plant lineages that have very fixed floral structures. So they don't have natural variation in the whorl numbers to start the investigation. On the other hand, of course, we know thousands of plant taxa that do have variation in their whorl numbers, but most of them do not have the molecular or genomic resources to push the investigation further. To this end, Aquilegia is really an ideal system that can help us further understand, can help us to fill in the blanks in our understanding. Besides the phylogenetic position, we have a well-sequenced and well-annotated genome. We have a molecular tools. We have variation in flora form. So it seems like the only thing that needed to happen is to have someone to study it. So in my dissertation, I sought to lay the groundwork for understanding how flora meristem termination is regulated in Aquilegia and thereby promote Aquilegia as a model system for studying this process, as well as other aspects of flora meristem regulation. This groundwork is laid by 3 main foundations. First, we ask how does the gene expression dynamics change during this window of flora meristem termination. Second, we ask how does the cell behavior dynamic change during this window. And thirdly, we ask what is the genetic architecture underlying this variation in this process. Before I jump into any results, let's get some Aquilegia Flora 101 out of the way. So here we're looking at the side view and the front view of a blooming Aquilegia flower. Gorgeous. The red and petal-like organs are actually sepals, and the long tubular-like organs are petals. There are 5 sepals and 5 petals. Inside of the petals, also sepals and petals initiate in alternate positions, as you can see in the diagram. Inside of the petals, most of the things we see are stamens. And to help us better visualize how do the stamens initiate, here we have 2 SEM images of 2 young flora buds of Aquilegia. The one on the left is younger. We can see a huge sepal here, and the dome of the flora meristem is still intact, meaning that this little flora bud is still in the process of initiating more organs. Whereas the one on the right is slightly older, it has all the organs initiated. That's why the dome of the flora meristem is gone, and I have removed all 5 sepals in that flora bud. Now, the organs colored in pink are petals. Again, there are 5 of them. For every Aquilegia flower, the stamens initiate in a whorl of 5 as well, but each whorl is tightly positioned against each other and they alternate in position just like the sepals and the petals. So there are always a few whorls of stamens that are positioned right above the petals, and then there will be a few whorls of stamens that are positioned right above the sepals, such as the one colored in blue here. Inside of the stamens, we will then have 2 whorls of organs that are called staminodes. Again, 5 organs per whorl, 1 positioned above the sepals, 1 positioned above the petals. Lastly, inside of the staminodes, we have 1 whorl of carpel, 5 in total. So you can see that compared to the 4 whorls of organs in Arabidopsis flower, our Aquilegia has way more whorls of organs and it's more complex. If you feel like you can't remember all the information on this slide within this second, don't worry about it, because I'll be constantly referring back to information that I mentioned here throughout the talk. And by the end of this talk, all of you will be experts in recognizing Aquilegia flora structures. Okay, so that concludes our Aquilegia Flora 101. Now let's come back to the big picture of this talk. So we want to understand gene expression dynamics, cell behavior dynamics, and the genetic architecture. Let's jump into the first one directly. In this chapter, I conducted in-depth RNA sequencing at 4 finely dissected developmental stages, which covered the developmental window before and after flora meristem termination. Each developmental stage, we had 8 biological replicates, and each sample is sequenced to an average of almost 20x coverage, which ensures the power and the accuracy of our analysis. Results from this project is broad, but it's very crucial because it tells us what is the transcriptomic dynamics in the developmental window, in the tissues that we're interested in. What genes are expressed and what genes are not? How does the expression of genes change over different stages? And what are the key genes marking the transitions between different stages? This result will serve as the main official directory of genes for many, many follow-up projects, many years to come. Subsequently, we're asking, how does the cellular behavior change during the window of flora meristem termination? More specifically, I am asking, how does the dynamic between cell division and cell expansion change during this window? Why is this important? Because almost every structure we see in a plant is achieved by the coordination between cell division and cell expansion, only these 2 processes exclusively. There's no cell migration, there's no programmed cell death. This is a very impressive fact if you also consider that all the cells in a plant are bounded by very rigid cell walls. So even for very small or similarly very simple structure, a tight coordination between these 2 processes is absolutely necessary. In addition, plant development is a continuous and dynamic process, and an in-depth understanding of any dynamic developmental process requires not only the information on the genes and the molecules, but we also need the precise geometric information of the cells, and we need them both in real time. So the previous chapter kind of touched on the component of genes times time, and this chapter is set to examine how does this geometric information of cells change over time. Traditionally, the examination of cells largely relied on histological sections or scanning electron microscopy. As we can see here, both methods can give us really good looking images, but they do have their limitation. If we're looking at cell scale, both of them lack resolution and they can only be applied to dead and fixed tissues, which is obviously not the most ideal way to study something that's dynamic. So in this chapter, I set out to develop a new method so that we can observe the developmental process in real time using a quantitative live confocal imaging. This project is in collaboration with Dr. Stephanie Conway, and we first of all developed a tissue culture protocol so that we can observe this entire process of the plant on a culture dish. Here on the leftmost panel, you're looking at a young flora meristem of Ecologia just being freshly dissected off the plant. And days go by, this little flora meristem underwent the process of continually initiating whorls of organs and then eventually the termination of flora meristem. Once we have the meristems growing on plates happily, we image them every other day, so every 48 hours. And we have 6 imaging points in total with multiple biological replicates. Since we're interested in the window of flora meristem termination the most, and we have a lot of organs in Ecologia, for this project, we mainly focus on the initiation of the last 4 whorls of organs, namely the innermost whorl of stamens, the 2 whorls of staminodes, and the 1 whorl of carpels. At time point 1, the innermost whorl of stamens are initiating. From time point 1 to 2, 1 whorl of staminode initiate. From time point 2 to 3, the other whorl of staminode initiate. And from time point 3 to 4, the initiation of carpel. So by then, we have all the organs initiated already. From time point 4 to 5, we focus on the morphological changes on the flora apex, which we can see the curvature is gone, become completely flat, and we call this period the making of flat star. This is my favorite developmental stage because it looks so incredibly cute. It looks like a star-shaped hat being worn by the flora bud. It's just so cute. Anyways, from time point 5 to 6, we enter the early developmental stages of carpels. And from the side view especially, we can see the elevated ridges of each carpel primordium. Okay, Now we have the images for all the time points. What can we do? Here we have an example of a flora meristem at time point 1 and 2. So the front view looks very similar, but if we pay more attention to the side view, we'll see that a whole new world of organ has initiated already. Then we can segment all the cells on these images using a software called MorphoGraphX, which looks like this. So if we look closer, you will notice that the cells of the same position are colored the same. And that's because all the lineages of all the cells has been perfectly traced already. So essentially, we're looking at the same cells at 2 different time points. If a cell has divided, such as those 2 at time point 1, their daughter cells will be colored the same as a mother cell to indicate the relationship. After lineage tracing, we can then quantify the changes happening in between, such as the number of cell divisions, the cell area expansion, or the direction of expansion. Since all these information represent the changes during the interval between 2 adjacent time points. So instead of have 6 single time points, our results will be presented as 5 growth intervals. And we have a few main focus for this project. First, of course, we're interested in the cell behavior dynamic change in the flora meristem. But we also paid great attention to the mechanisms of primordial initiation in early development, and we compared the early growth between different organ types. Due to the limit of time today, I will only share part of the result at point 1, and then I will give you a sneak peek of point 2 because they're focusing on very different things. And hopefully them combined together can show you the power of our analysis. Okay, so for the first part, if we want to present growth of these 5 intervals, we will see something like this. I know, suddenly there are a ton of new information on this slide, but don't worry, let's break it down. Firstly, we can see every cell is colored, and this color represents the changes of cell area. If a cell is dark or black, it means that its size did not change or even become smaller during this interval. But if a cell is bright yellow, it means the size has increased at least by twice at the later time point compared to the previous one. And we can also see some of the cells have their outlines highlighted in white, and those are the cells underwent cell division during this interval, okay? Now we focus on the regions from the center of each flora bud to the edge of the latest world of initiating primordial. We normalize this distance from the center to the edge, and then we divide them into 6 equal bins, even bins. In this way, we can not only compare the growth dynamics between different intervals, we can also compare the dynamics between comparable regions of the flora bud based on the bins. On the left, we have the cell area expansion plot, and on the right, we have the average cell division plot. And the color for each interval is indicated below them. For now, let's focus on the first 3 bins. Why? Because these are the regions represent the centermost part of each flora bud, and that's part of the flora meristem before the termination process happens. If we look at the cell area expansion map on the left, it doesn't take us long to realize that there seems to be a clear distinction before and after time point 4. From time point 4 to 6, the cell area expansion is much lower compared to all the intervals before them. In fact, we see the same pattern for the average cell division, that from time point 4 to 6, the average cell division is much lower, especially in bin 1 and bin 2. Okay, so now it seems like we have some really simple, very straightforward results that we see a clear distinction between before and after time point 4. So what? Well, it's actually kind of a big deal because we would not have anticipated this at all based on what we know in Arabidopsis. At time point 4, the carpal primordia has physically initiated already. Look at those shoulder pads there. Those are the carpal primordia. And based on what we knew in the comparable stages in Arabidopsis, the decrease in cell division, the decrease in cell expansion in the center should have happened before, not after the physical initiation of carpals. In addition, if we look closer at the bin 1 of the cell division plot, we can see the interval between time point 3 and 4, the pink one, had a slightly yet very significantly higher rate of cell division in the centermost part of the flora bud. This increase in cell division at this stage is very interesting because it really challenges the conventional notion that termination always happens before the physical initiation of carpels, and once it happens, nothing will be going on in the center for a while. Now that we know Arabidopsis and Echologia are different, maybe Echologia is a weird one, maybe Arabidopsis is a weird one, we don't know, but we'll have to investigate further. Also, it's kind of amusing to say this out loud, but our result also highlighted the importance of having more than one data point of studying a given developmental process. Okay, so this is the first little piece of result. And now let's turn to something very different for just a minute. So here we're looking at the same primordium at time point 1, 2, 3. And we can see this little young primordium is gradually bulging out. In order to really analyze in detail how does a primordium grow, we can label some cells. So here we have 10 cells outlined in blue and 10 cells outlined in green. In order to see visually, physically see where these cells are, we can use a curvature heat map. So in this heat map, we can think of cells that are colored in red and blue are curving towards the opposite directions. So all the cells outlined in blue have positive curvature, they're bulging out, and they made up the majority of this young primordium, whereas cells in the green outline, most of them have a negative curvature, they're in blue color, so they concave in, they're in the boundary region between this young primordium and the primordium located below it. Through lineage tracing, of course, we can label all the cells on different time points and quantify the change. For instance, here we're seeing the cell area expansion map again between time point 1 and 2. And if we outlined our cells on this heat map, well, isn't that so interesting? Only the cells outlined in green showed the highest growth during this interval, whereas cells outlined in blue did nothing impressive at all. Besides cell area expansion, we also looked at cell division and the cell expansion directions. But long story short, let's come back to the end to see where those cells end up. At time point 2, we can already see that the cells in the green outline made up about half of this primordium already, while at time point 3, the cells outlined in blue, half of them end up in the boundary region with negative curvature, whereas cells outlined in green became the majority of this primordium. So what we're seeing here is something really interesting. It seems like we almost saw a displacement of cells during this initial stage, that the cells originally made up the majority of this primordium did not stay as a majority of the primordium. In addition, our result also revealed that this initial primordial growth is mainly driven only by a single layer of cells that located as a lowermost position of an emergent primordium, which really begs the question of what controls the growth in those cells. So I hope you're entertained by these two small pieces of results, and there are a lot more results that I didn't get to share today due to the limit of time, but we'll have 2 preprints dropping very soon in a week or 2. One will present the detailed analysis for all the growth, and the other will present a detailed protocol about how to conduct live imaging in Echologia and how to reproduce all our results. This project is the first time that anyone looked at meristem dynamic and primordial growth in Echologia, of course, but it's also the first time that the flora meristem termination process being characterized quantitatively at a cellular level. It's the first quantitative live imaging in a non-conventional model system, and our method is likely applicable to other non-conventional model systems as well. We're super psyched about this project because it's an important step that will make monitoring gene expression in real time possible. And eventually we will know how do the expressions of the genes controls the precise interactions, mechanisms of cell division and cell expansion in real time, and which directly led to development and morphogenesis. Now, after looking at the broad dynamics of gene expression, broad dynamics of cell behavior, let's talk about genetic architecture and genes. In this chapter, we use QTL mapping as an approach to proactively look for candidate genes. QTL stands for quantitative trait locus mapping, and normally it starts with two parental individuals with different phenotypes. Through the solving of F1, we will have a population of F2. The genomes of the F2s will have different fractions of genomes from the parental individuals. And through assessing the phenotype and the genotype of each F2, the correlation between the variation will tell us what is the genetic architecture. And for our purpose, of course, we're interested in the genetic architecture underlying flora meristem termination. In collaboration with Dr. Evangeline Ballerini, who crossed two sister species in Aquilegia, Aquilegia brevistyla, Aquilegia canadensis, which give us some perfect intermediate flower, F1 flowers, and a whole population of F2 that looks like this. I know, very stunning, but please don't get distracted by the shapes and colors for now. I want to remind you that I want to use this population of F2s to map a process that we know nothing about. Well, now you can ask me that you just spent most of the talk telling us flora meristem termination happens at the earliest stages. How the hell are you going to map it in the mature flowers? Well, great question. It turns out our flowers are very good to me because they have the same flora structure except for stamen whorl numbers, which really provided us with an ideal opportunity. Let's think about it. If everything else stayed constant, and the only variable is the stamen whorl number, that means the differences in the stamen whorl numbers can be directly translated into the differences in the timing of flora meristem termination. If a flora meristem is terminated early, fewer whorls of stamens in that flower, but if the flora meristem stayed active longer, more whorls of stamens in that flower. In other words, I'm using stamen whorl number as a proxy, as a quantitative trait to represent the timing of flora meristem termination. In other words, I'm counting stamen whorl numbers in flowers, and here's how I count it. Just a reminder, we always have a few whorls of stamens that are positioned right above the petals, and then a few whorls of stamens positioned in between the petals or right above the sepals. Here we have the SEM images of 3 F2 flowers. If we count the stamen whorl number that are positioned right above the petals, we have 3, 3, 4. If we count the stamen whorl number that are positioned in between the petals, we have 3, 4, 4. So for these 3 examples here, their total stamen whorl number would be 6, 7, and 8, respectively. In total, I have counted almost 5,000 flowers from more than 400 individuals. And if we take a broad overview, we can see the mean stamen whorl number for the parental species are nicely clustered at the lower end and the higher end, and the F2s spread across the entire range. For each F2 that we phenotyped, we also conducted whole genome sequencing for genotyping and constructed a genetic map that looks like this. So now with the phenotype and genotype in hand, I can finally do QTL mapping, and the end product looks like this. Here on the x-axis, we have the 7 chromosomes of Aquilegia, and on the y-axis, we have the lot score, which is an indication of likelihood. Anything above this blue line will be considered as a real significant QTL, and voila. We got QTL. More specifically, we got a lot of QTL, 1 on each chromosome. This tells us the genetic architecture underlying the trait of interest is complex. It's controlled by many genes together, potentially with the interactions between some of the genes, and each gene controls a small effect. All the QTL together can explain about 50% of the phenotypic variation. Then I narrowed down the genomic regions that contains the real QTL and started to look for candidate genes with the help of the RNA sequencing data. Specifically, I'm interested in genes that are expressed at early developmental stages. I'm interested in genes that are differentially expressed pre and post to flora meristem termination. And I'm also interested in genes that have their homologs being functionally studied before and have any kind of meristem related functions. With these criteria in hand, I got my first list of candidate genes that I'm super psyched about. Now, let me give you a small example of one of the genes, little zipper on Q2. So here we're looking at how little zipper, this gene of interest, is expressed during the early developmental stages of flora meristem. The cells that are stained brownish purple are the cells expressing little zipper. So we can see that little zipper is strongly expressed at the organ boundaries. This is an expression pattern that has been previously observed, also functionally studied in both Arabidopsis and tomato, indicating a functional conservation in our flower as well. Then I want to bring your attention to those regions where little zipper is also expressed, although not as strongly. So these are the regions overlap with where the stem cell population are in the flora meristem, and this expression pattern has also been previously observed in some vegetative meristems. Lastly, please look at those regions which we have strong specific expression of little zipper, and this expression only across the first layer of cells through the flora meristem. This expression pattern has never ever been observed before in any other system, and so specific here really makes us wonder, what is this gene doing in Aquilegia flora meristem? So this is a little example of one of my candidate genes. This list of candidate genes is in fact the first list ever that can potentially tell us something about how is the variation of flora meristem termination controlled. And we're able to get this first ever list of genes because we have this amazing system in our flower that everything stayed constant with only one variable. This question simply cannot be investigated by any of the current model systems because they don't have such variation in world numbers. Obviously, we're super psyched about this project, and again, a preprint dropping in a week or two. Now is a good time to bring back your attention to the bigger goal of this dissertation. So today, obviously, I cannot tell you who are the genes and how do they interact to control flora meristem termination in Aquilegia. But at least in my exploratory dark space, I know the size of the space, I know where the floor is, I know where the ceiling is, and I have some promising directions to point my flashlight. So I know I have been talking nonstop for almost an hour, and once this presentation closes, you will forget most of the things. But I did prepare 3 little take-home messages if you're gonna take home anything with you. First, plants are awesome, in part because of meristems. Second, Aquilegia flowers are awesome and gorgeous, and they are important model systems for flora evo devo studies. Lastly, flora meristem termination is an important developmental process in making a flower, and I have tried my best in the past few years to push our understanding a little further. So before I close this presentation, I would like to thank a lot of people who made this thing possible. Firstly, I want to thank my department, my school, and my professional societies. So for the past few years, I have been very vocal about my resentment that international students studying in the US don't get equal chances for awards and fellowships due to citizenship requirement. Every year when the big fellowship and award application seasons open, such as right now, it always felt like that we are just standing by a gate and looking through a festival going on in town, but we're not allowed to enter. It is one thing that whether someone applied to award, whether they get it, it's another thing that whether someone is allowed to apply. So I have been very bitter about this, not just for myself, but for everyone. However, during my PhD, I have been fortunate and extremely privileged to have all these organizations support my dissertation, and without which my dissertation probably will look very different. The merit fellowship from GSAS relieved me from teaching duty and give me time to finish some crucial experiment when there's literally no other big fellowship that I'm eligible to apply to. Chapter 1 is entirely supported by the Student Research Fund from OEB, and Chapter 2 is entirely supported by the Emerging Model Grant from the Society of Developmental Biology. As for Botanic Society of America, besides all the research and travel awards, it gave me more than I can ask for. It gave me a community and all the skills I would never otherwise learned during grad school. So I served on the board of the society for 2 years as the student rep. And during those 2 years, I learned to organize literally any kind of event you can ever imagine, and also every little detail of how to run a professional society, from membership to management, to accounting, to publication, to investment, to PR, to release statement, everything. Many staff members and people who serve the board with me are here today too. And I want to thank each one of you for all the things that you taught me through those 2 years and onwards. And I have benefited so much from all the professional societies, and I'm ready to give back through the rest of my career to make our societies better so that we can help reach more people. I want to thank my committee, Cassandra, Ned, Terry. You guys are the best. You're awesome. I thoroughly enjoyed my qualification exam. I thoroughly enjoyed every DAC meeting and every private meeting with you. Sometimes I cannot believe how lucky I am to have a committee that understands my goals, that help me to navigate through all the unknowns, and you really celebrate all my progress as well. I am so lucky, and thank you for all your support and guidance. I want to thank my undergrad mentor, Fujita Sensei, who's also here today. And I first joined the Fujita lab when I went to Japan in my junior year. And Fujita Sensei really taught me everything from the smallest things, from how to pour a gel in the lab to how to be a scientist. I have to say that at that time, I was not an easy student to teach because I have never spoken English before. That was the first time I spoke English daily. And I didn't know how to say most things related to lab. And I couldn't speak any Japanese because my program was supposed to be in English. So I was really hard to communicate, but Fujita Sensei spent so much time with me every day. He has been so patient, and he cultivated my curiosity in science. I always have absolute confidence in my wet lab skill because I was trained by Fujita Sensei. Sensei, 本当にありがとうございました。私はいつも頑張っています。I want to thank the Kramer Lab, who treated me very well for the past 6 years, and we had some stellar undergrads working in our lab. Special shout out to Claire, who sensed my complete ignorance but a slight curiosity about American culture when I just arrived. She arranged almost weekly movie night that we went through all the classic high school movies in the 90s and the 2000s. Those are the movies I will never watch on my own, but it was really fun to watch as a group. And now I'm an expert in American high school cultures, I guess. And thank you to Steph. Oh, shit. I just cursed in my defense. Thank you to Steph, who I first met in a botany conference. It was very weird and magical because I talked to her for 5 minutes and I got the feeling that I know her for 5 years. More magically, Steph joined our lab afterwards as a postdoc, and it's just magical onwards. It was such a great experience to collaborate with you and be your friend, and I just enjoy every minute I spent with you. Since we're talking about Kramer Lab, so thank you also goes to Angie, our Kramer Lab alumna and collaborator. It was really fun to go to California with Molly to meet you, and we had an awesome collaboration going on. And of course, to Molly, my lab twin. Molly and I started grad school together at the same time, but we are very different people. Molly and I have very different personalities, so either you will be good or bad, right? But the difference between us became the strongest strength that we have for supporting each other. We have spent so much time together. We know each other so well. Sometimes it's crazy to think about. Molly knows exactly when I'm annoyed in a given situation, even if I don't do anything, I don't show anything. And I know the consistency of egg yolks that Molly likes. I also know right now, Molly kind of wants to cry, so don't look at her. I want to thank my cohort. And the entire OEB probably is very tired of us saying, we're the biggest cohort in the history. Well, we are, and it's different. The amount of community and support we have is different. I enjoyed my grad school very much because all my friends, and it was really fun to go through all the photos, and it was very difficult to decide which ones to put on this slide. We literally have been doing everything together, been to all the places together, and it's magical. Special shout out to Brandon, because he's probably the only person who knows approximately how many times I failed my experiments. Brandon and I played music together for 2 to 3 years until COVID, but surprisingly, we never took any picture together while we were playing music. So I guess we never played music together. Anyways, thank you so much for listening to all my complaints, big and small, and also share all my joys. Thank you to Crystal. Crystal and I started our master's program together. Then we moved to the US together to start grad school. So Crystal has always been a very constant presence in my life. And I could not have imagined an alternate timeline that I didn't come to grad school with Crystal. You're an amazing person, you're an amazing scientist, and I'm so proud of you. So Brandon and Crystal knows that they're special to me, and sometimes they get into fist fights to see who is my favorite. And this is one of the fights that I caught on camera, and look at how invested everyone is. Look at Dan, look at Amani. I will not tell you who won this fight to protect their privacy. And thank you to my friends that we met in Europe, and I love and miss every one of you so much. We always met at least once per year in a different country, but because of COVID, it has been too long. And I can't wait to see you guys again somewhere in the world. Thank you to my husband, Wei Lin, who has been a source of joy, a source of encouragement, and source of inspiration. He really did all he can to support me through this journey. And I think you're great. And it's good that you think I'm great too. So we'll see what happened to us next. Since I talked about husband, of course, we'll mention our cats, Penny and Nickel. I was not a cat person at all until these two cats entered my life. And the amount of love and encouragement and comfort they give me during all the downtime, during the entire COVID lockdown, during the dissertation writing, is beyond words. I love them so much. Pets are the best. And Elena. The amount of support and help Elena gave me is beyond words. I don't even know where to start. But one place I can start today is that during this presentation, I have said a lot of I statement. I thought about this, I want to do that, but most of them is a lie because every idea I have received help from Elena. Elena helped me to develop, to shape, to polish all my ideas. She helped me to become the best scientist that I can ever be right now. And Elena is not only a mentor that I feel I share all the honor with. I feel comfortable to tell her I'm tired, I'm exhausted, I don't want to do this, I completely messed up. So Elena taught me how to science, how to teach, how to mentor, and she gave me incredible imposter syndrome since the beginning of the grad school, because I was like, if that's the standard that I have to reach to be a professor, I would never be a professor. Now I'm leaving grad school thinking exactly the same, but one of the things I learned for the past years is that I will fake it until I make it, and I know she will always have my back. And lastly, my parents. I have left home for many years already, about 10 years. And normally I go back home once per year and I stay for 2 to 3 weeks, 3 weeks maximum. So it's not a lot of time already. But recently, due to teaching, due to COVID, the last time I saw them in person was 3 years ago. During these 10 years, my parents have endured a tremendous amount of loneliness because I'm the single child, and also a lot of doubts and questions from other people. So what, your daughter goes to Harvard? She's not here with you when you're sick. I wouldn't want a daughter like that. How much money does your daughter make? What? From Harvard? That's so much less than how much my kids made right out of college. Why is she doing a PhD? That will really deter people. Why is she not married? Why is she not having kids? She's passing her prime. These are real things that people have the audacity to say that to their face and make them feel miserable. What do? They bear those crap on their own. They understand that I want to do something, they let me do it and support me. This sounds like a very small deal, but no, it takes courage. As you can see today, I put a lot of Chinese on every slide so that for the first time, my parents can understand what I do for my research, but they wouldn't understand anything I'm talking about right now. However, I know during the past hour, they couldn't be happier, they couldn't be prouder, and that makes me very happy and proud of them as well. So everyone, please do me a favor. Please go to Google Translate and say something to them in Chinese in the chat. Traditional Chinese, simplified Chinese, it doesn't matter. Can you just let them know that you know they are here? And if you can do that, I really appreciate it. Thank you very much. And with that, I conclude this presentation, and I'm happy to take any questions.
Big applause from everyone. That was awesome. Okay, so we can take some questions. We can take a little time, take a few questions. That's okay.